Welcome back, life science learners. Trust that you've been able to refresh yourselves. Let's get back into our lesson. Again, we're spending some time revising the basic concepts around DNA as a molecule and its importance as a code of life. We've discussed some important terms and concepts such as DNA replication as an overview. We've looked at protein synthesis as an overview. As I mentioned, it's important that you prepare some notes on these and understand how these processes occur. They're very important in terms of preparing for a question on an essay where you need to describe DNA replication or, for that matter, protein synthesis. However, in this segment, we're going to spend some time looking at how we apply our understanding around the structures of DNA and some concepts around how DNA replication occurs. So let's get straight into it. I encourage you to pull out a book, and if you can write down some answers as we work through them, I'm sure we can check these as we get through the lesson. So the first question, if we look at it, the diagram alongside represents a portion of a DNA molecule. So it tells us that it's a portion of a DNA molecule. If we look at it, we can clearly see that it's got two strands on either end, and there's some base pairing that's occurring right here in the middle. So we do know that it is DNA as it's mentioned in the question, so we don't have to figure out whether it's a DNA undergoing process called what we refer to later on as protein synthesis. So this is just a simple DNA molecule where we see it's double-stranded. It's important that we recognize the different labels. And I often encourage the learners when they see a diagram to try and understand what are the different parts before getting into the question. So I can clearly see that there's an area that's circled having three different parts. We can see a structure here that's labeled B. We can see a structure labeled C and D. So let's get straight into some of the questions based on this. So as anticipated, identify B and C. So guys, if we look at structure B and if we look at structure C, we can clearly see that it is pointing specifically to what we refer to as B points to the complementary base pairs. Now, obviously this is a DNA molecule, so that's going to be a DNA nucleotide that it's pointed to specifically. When we look at structure C, we got to recollect that DNA is double-stranded and it's made up of a sugar phosphate backbone. So here we find the sugar molecules, and here we find the phosphate molecules that form the sugar phosphate backbone. So we know that in one single nucleotide you have a nitrogenous base, we have a sugar molecule, in this case it's deoxyribose sugar, and we have a phosphate molecule here. So molecule C points to a phosphate molecule, and molecule, as I mentioned, B in that previous diagram pointed to a nitrogenous base. Okay, let's continue with this question. A, 1.2, monomer A. Now guys, the term monomer is another word for the concept of building blocks. Now remember that DNA is made up of building blocks, which we refer to as DNA nucleotides. And those nucleotides consist of three different parts, as I've mentioned in this illustration. It contains a sugar, it contains a phosphate, and a nitrogenous base. So the building blocks, or another word for building blocks, is monomer. In this diagram, monomer A is a DNA nucleotide. And I've got to be specific because this is a DNA molecule. Often we get learners simply replying, it's a nucleotide. And I think it's important that at this point that you're quite specific in being able to differentiate that it is a DNA nucleotide and not just a nucleotide, because it could represent an RNA as well. So the correct answer would be a DNA nucleotide. And it's important that you are able to express yourself correctly and scientifically right. Let's move on to B. Two scientists who received a Nobel Prize for the discovering of the DNA molecule. Guys, it's important that we recognize that DNA was discovered collectively over a period of time in different stages. However, there were three significant scientists that contributed to the discovery, and we go back into the history of that. We know that Watson and Crick, so the fellowship between James Watson and Francis Crick, were recognized for that, obviously, when they put together the model of the DNA. We also know that Rosalind Franklin was posthumously recognized for her contribution to the discovery of the DNA. So there were three significant scientists that played an important role: James Watson, Francis Crick, as well as Rosalind Franklin, in terms of her process of being able to crystallize DNA and photograph that. So again, you've got to be able to identify this. It's important that we recognize scientists in their endeavors and we recognize them in terms of their contributions to science. The next question: one organelle in a cell where DNA is located. Guys, it's important that when we revise DNA, we recognize that DNA is found in different structures. For example, we've been looking at the nucleus. However, we do know that DNA is also found in the mitochondria, and it plays an important role in how some of the genes in the mitochondria are expressed. We also know that plant cells in their chloroplast contain some DNA, and that DNA plays an important role in the expression of those genes or those producing important characteristics in those specific organs or organelles rather. So let's recognize the importance of these. So we mentioned nitrogenous bases as being structure A. We also mentioned that these individuals, James Watson, Francis Crick, Maurice Wilkins, as well as Rosalind Franklin, much later was recognized for that. The organelles in which the DNA can be found included the nucleus, the mitochondria, and in plant cells, the chloroplast. Let's move on to question 1.3. Describe how a mutation on DNA may change the structure of a protein. And that's a 4-mark question. Guys, it's important that we recognize that mutations, again, the definition of a mutation is a change in the sequence of a nucleotide, of a gene. And that affects a single nucleotide, or it could affect more than one nucleotide. So DNA is often affected by mutations which the cell generally corrects. However, sometimes these mutations go by uncorrected, and that we refer to as an alteration in the sequence of a gene. So let's try and unpack how you would answer this question. We've got to recognize that genes code for the sequence of amino acids that make up a protein. So when there is a change in the gene, it's going to in turn affect the chain of amino acids. Well, it may affect, depends on the type of mutation that takes place. So let's try and unpack how you would answer a question like this. It's important that we recognize that, I'm going to change my pen so it makes it easier for us to differentiate, that DNA will code for characteristics. So when the DNA changes, and that happens because of an alteration or a mutagen, we say that a mutation has taken place. This leads to a different mRNA being produced or a change in the codon. So we do know that when DNA is being copied into what we refer to as mRNA, if there's a mistake or a problem in the process of it being copied, and that's mRNA, if there's an alteration in that, or if there's a mistake that happens at that specific point, it in turn, when it leaves the nucleus, carries what we refer to as an abnormal or a problem in this. So that could be an altered nucleotide on the mRNA. And this means that this entire codon has now either changed or could be coding for the same amino acid. So we refer to that as a different mRNA being produced, having a different code. Okay, this will match with a different tRNA and produce an anticodon that does not carry the initial amino acid that was coded for by the unaltered codon. This results in a different amino acid being brought by the tRNA to the ribosome. In turn, this leads to a different protein or an altered protein. So guys, we do know that mutations can be neutral, where a mutation like this sometimes codes for the same amino acid, and we refer to those as neutral mutations. We also know that certain mutations can be harmful, where it affects, in this case, an important protein. And we know that mutations like sickle cell anemia, which affects the shape of the red blood cell, is a harmful mutation. We also know that sometimes certain mutations can be beneficial, where it creates a protein that gives some individuals an added feature. For example, there are some populations in the world that have a gene that is mutated and made them resistant to HIV. So again, that's a beneficial mutation if one has to look at the benefits of being resistant to HIV. So again, we need to recognize that mutations have both neutral as well as harmful or beneficial effects. And that's essentially an answer to that question. Okay, the next part would be tabulate two structural differences between a monomer of RNA and a monomer of DNA. Guys, the key words are structural differences. So it's important that when we compare the differences is that we talk about the structure. We cannot talk about the location of where the DNA is found in RNA because that's about the location. Let's reference the structure in that specific answer. So you could answer by describing that mRNA contains a ribose sugar, whereas DNA contains a deoxyribose sugar. We also know that in mRNA you have adenine, uracil, guanine, and cytosine. So we talk about AUGC, whereas in DNA you have your adenine, thymine, guanine, and your cytosine, where thymine and uracil are the two bases that are different. Okay, so let's move on to the next question and let's apply some of our understanding further on to this process of protein synthesis. So the diagram below represents a process that occurs during protein synthesis. Now guys, remember that as I mentioned, protein synthesis happens in two parts. The first part is what we refer to as transcription, where in the nucleus an mRNA is produced. A transcript of the gene is produced as mRNA. That then moves into the cytoplasm, where we know the process of translation occurs, which is putting together a chain of amino acids with information that comes from the mRNA. So here we're looking at this illustration that shows you these ribosomes. And these ribosomes are often made up of two units, and I often like to use the word the snowman. And if you remember that, you can easily identify what we call a ribosome. So here we're seeing a ribosome, two of them actually reading through a strand of mRNA which has these codons on them. We also can see that there's some tRNA molecules onto which unique or specific, I'd like to refer to them as amino acids attached, which you can see here. And these are based on the codon and anticodon base pairing. So that's an overview of this illustration. And let's see some questions on this. So the question is, identify the process above. And if we look at the process above, guys, as I mentioned, this process is certainly happening at the ribosomes outside in the cytoplasm. So that process again refers to the process of translation, where we see a chain of amino acids being produced, and that again is a key part of the final product of translation, where we find the amino acid sequence put together. Okay. Let's look at the next question. Identify organelle A. So if we look at organelle A, as I mentioned, guys, it's a structure that is important in protein synthesis, and we refer to that as the ribosome. And I often remind the learners to think of a structure that's made up of two units. It's got a small subunit and a larger subunit, which is often a unique feature of these ribosomes. They're three-dimensional structures, guys, in which the mRNA moves through. And that's essentially what is being illustrated in this diagram. The next structure we need to identify is molecule B. And as I mentioned that during the process of transcription, we find that mRNA is produced, and that mRNA leaves the nucleus and moves into the cytoplasm, where the ribosomes read that. So clearly we can see that the ribosome is processing this mRNA molecule. Several of them read this mRNA molecule and then bring in a specific tRNA with its amino acid to it. The next question is bond E. Okay, so we know that amino acids put together form a polypeptide, meaning a chain of amino acids. Often we ask, what are the bonds that hold these building blocks together? And learners often tend to confuse the term polypeptide chain and polypeptide bonds. Unfortunately, you've got to mention that it is a peptide bond that is found between your amino acids and not a polypeptide bond. That's the polypeptide refers to this chain of amino acids that make up the final sequence of amino acids that form in that protein. Okay, so as I mentioned, that's a quick check of our answers. And as we wrap this up, let's very quickly look at one more question. Give only the letter of the molecules that carries the amino acids. So letters that carry the amino acids, if we look at it, This would be a structure that's carrying the amino acid, so it would be letter C, and that structure is referred to as the transfer RNA. It's copied from DNA. We know that mRNA is copied from DNA, as I mentioned earlier on, so that's going to be structure B. And finally, is this monomer or building block of protein? So is the monomer or building block of protein? And so we know that these are the monomers or building blocks of proteins, and that would be structure labeled D.